You can support the Double Loop Podcast by contributing at patreon.com/Double Loop Podcast. Thank you to our supporters, and we hope you enjoy the show. Welcome to the Double Loop Podcast, your source for everything about fingerprints. While you're working on your comparisons, we'll talk about comparisons. I'm Eric Ray.
And I'm Glenn Langenburg.
Hey, Glenn, what do you call a Vietnamese soup with imitation ingredients?
Fuck.
Close. How about a faux pas?
Okay. Good one, Eric.
Thank you. I made that one up myself.
Oh, that's why it's extra bad.
Right. So, Glenn, what do we have to start off with today?
Well, I just wanted to read a quick email. We actually had a really nice email, so a quick shout out to Ava. Ava is in New Zealand, and actually I met Ava when I was in Auckland for a conference, and she wrote a very long email about spending some time. She was a woman that drove me and ETL around in Auckland and the countryside, and a couple other people. And she was just relating about how she was listening to ETL and I go back and forth very vigorously, talking about cognitive bias. I probably said it was BS, and he got very upset. No, I'm kidding, of course. Error rates and likelihood ratios, and she was still, in training or just starting her training, and was really just learning this and listening, and was just talking about how it imprinted on her. But then she gets into a very nice part of her email that says, since then I've been binge listening to the Double Loop podcast, which thankfully pointed me to exploring and acquiring some very interesting knowledge. And then, I try to absorb a lot of this. I'm not sure that other people in my agency are as interested in talking about the episodes or, sharing this, but I continue to listen to it, and I just want you to know that your podcasts have changed my perception on things in relation to fingerprint evidence in a very good way, and I hope that you will continue to do this. Thank you, Glenn, and please pass on my appreciation to Eric. Your podcasts have opened my mind, and I have been just overwhelmed with all the things that you guys have talked about. And she continues to gush for several more paragraphs, which is very flattering. And I just thought it was really nice. And I just wanted her to know, thank you. That it's a really nice sentiment going out to Ava in Auckland and New Zealand. And these Kiwis, they're so nice. And it's nice to hear such positive feedback from listeners, knowing that we make their day and that we give them lots of good information for them to go back to and learn about. I thought it was really nice.
Yeah, thank you, Ava. Like we talked about last time, maybe that's the moment of zen for all those examiners, so that they can clear their mind and reset and refocus. What was the technique? Mindfulness. Maybe hopefully this is our moment of mindfulness. No, that's really great to hear. Thank you, Ava, for passing that all along.
It was the Daily Show that used to have here's your moment of zen.
Oh, your moment of zen. There you go.
I always liked that. It was always such a nice sign off. Here's your moment of zen.
Yeah. So thanks again, and as long as everybody keeps listening, we'll keep making them. And it just sounds like we're just reaching more and more examiners, and that really is a great feeling. So
yeah, absolutely, around the world. So Eric, today we have a guest with us. I'll introduce you in a second, but if listeners might recall, a couple episodes back we had Dr. Niki Osborne on, and or as they call her in New Zealand, Niki. We had Niki on, and from the block, Niki from the block. And she was on, and she had recommended that we speak to Theresa Stotesbury. And so today we're going to have a guest, Theresa, on here. And Niki had told me a little bit about her research when I was hanging out with her, and I was pretty fascinated by it. One of the things in my background is bloodstain pattern analysis. And in fact, I really enjoy it. There's a part of me that wishes I was doing it more often and could still get involved. I think if I ever went totally private, I would definitely get back into that because I do find some really fascinating cases. And
Right.
so some of the stuff that we're going to be able to get into today deals with that because Theresa was involved in developing some pretty cool stuff that she's going to talk about. So I'd like to welcome Theresa Stotesbury. She's at Trent University, which is in Petersborough, Ontario. And where did you say that was, Eric, in relationship to
Uh,
Toronto?
a bit northeast of there, not quite halfway from Toronto towards Ottawa. Is that right, Teresa?
You bet.
All
Oh,
right.
all
Yeah,
right. Well, let's
halfway
welcome
between
Theresa.
Toronto and Ottawa. Hi, guys.
Hi.
Thanks so much for
Hello.
having.
Yeah, thanks for joining us here on the podcast.
This is really exciting.
Well,
Well, that's
we
great
are
to hear.
we are very excited to have you, and Nikki just talked up your research so much. She said such great things, so I'm very excited for the listeners to hear about it. And I know it's not traditionally a fingerprint thing, but occasionally fingerprint examiners do get involved in a little bit of blood blood patterns in the sense that bloody fingerprint impressions are bloody transfers. So they really should, in my opinion, know something about the mechanics of blood and blood transfer
Yeah.
because it does relate when it comes to distortional issues and interpreting bloody friction ridge impressions. So it's good for them to get a little bit of exposure to something slightly outside the fingerprint realm. So welcome, Teresa.
And it, so Teresa, I'm not sure if Glenn saw it, but earlier today, like one of the last emails as we were kind of arranging things, you sent a photo or a scan of your own bloody fingerprint of a double loop whirl.
Like, how perfect was that, guys?
That's
Exactly.
awesome.
Right? Yeah, it was quite interesting. I had this month of mayhem where I was on the road at various conferences, and you know how it is, trade shows and everything in between. And I was scanning my emails, and I got an email from a forensic podiatrist from somewhere in the UK, and she was asking me, "Oh, can you make impressions with your fake blood?" And I thought, well, that's really interesting. And I got thinking. I had a day in the lab about impression evidence and you know what we could do with the synthetic. And I was looking at my fingerprints, and I was like, oh, well, I definitely have a double loop here. This is kind of perfect. So I had some fun in the lab and made some fingerprints for you guys.
Yeah,
Fantastic.
the images were very cool. So in your introduction there, you tipped your hand a little bit. So let's discuss. Let's jump to your research and what was it that Nikki said that I should be talking to you about, and you should share with our listeners. Why don't you tell listeners about what you developed?
All right. So I make fake blood or artificial blood. I guess we call it forensic blood substitutes, synthetic blood substitutes, tomato, tomato. You may have heard it in some sort of capacity in the forensic world. But what I like to call it are forensic blood substitutes because we make fake blood that you can't put into your body. And we make them for the purposes of research and training and crime scene reconstruction.
That is pretty cool.
Huh.
What can we? I mean, without getting to proprietary stuff, what are we talking about? What's it sort of made out of?
Yeah, no, that's fine. So the material is proprietary or trade secret, we call it. But
Nice.
the basics, yeah, the basics of the chemistry is it's a silicon sol gel material, which is basically just how I like to describe it as a polymer that forms a liquid glass.
Mm-hmm.
So you can make stains or any sort of blood stains, and eventually the stains will dry and form a thin film on some sort of substrate, and make produce kind of the patterns that you, I guess, want to use for your research or your training or any sort of course material.
Okay, so
Interesting.
I have tons of questions for you. Let's start with let's start with the first one related to it. Obviously, anyone who knows anything about blood, as it follows these non-Newtonian mechanics and very specific mechanics and fluid dynamics that blood has. I assume that somehow you're able to mirror that in some way.
Yes, actually, I spent the majority of my PhD looking at the fluid mechanics of blood and making artificial fluids that do the same thing. So blood is a non-Newtonian fluid, meaning, and it's more specifically a shear thinning fluid, which means that the more shear force and the more force that you kind of push onto blood, its viscosity decreases with the more force that you push onto it. So it's
You
quite
explain that
unique
really
because
well.
oh, thanks.
No, I mean
Huh.
I never hear people explain it so well. I mean, they start getting to the physics of what Newtonian and non, but you explain that really really well. That's fantastic.
So
Oh, well, thank you.
the non-Newtonian, usually when I hear that phrase, it's that that gooey stuff that when you push on it, it gets viscosity gets greater. So like cornstarch in water. I've seen like people work with that, and it seems like when you like punch it or push it real hard, it hardens up and becomes more of a solid at that point. You're saying blood is like the opposite?
Right. Yeah,
Okay,
it is the opposite, and so you can imagine blood is a complex fluid, and it contains lots of different components, like different cell types and different proteins and things
right,
like that in the blood. And really low shear, like really low force settings, the red blood cells they actually stack on top of each other, and you get this kind of phenomena called a rouleau, and so that has a really high resistance to any sort of force that you push on it. So it has a really high viscosity. But then as soon as you give it just a little bit more force or more energy, the red blood cells will disperse and then start flowing. The viscosity will decrease and then kind of plateau and flow more Newtonian-like, I guess, where you get this like nice little plateau region where the viscosity stays relatively constant, and that's in pretty high shear conditions. So, and I guess in our case in forensics, in relatively, I guess, violent situations.
So
huh, interesting.
when you were studying the dynamics of it, obviously you're doing certain kind of bloodstain patterns you're probably creating. I assume you were comparing them to actual blood and probably looking at some of the differences, and I assume that they were negligible in some way.
Yeah, this is actually it's quite interesting that you say that because at Trent and in a lot of universities and even some labs around the world and in Canada, it's really difficult to actually use human blood and whole human blood in your
Mm-hmm.
testing.
Right.
So we use our kind of analog in Canada is sheep's blood. We get it sourced from the Canadian Food Inspection Agency.
Mm-hmm.
No sheeps were harmed in the process, which
Really?
is nice to know.
That
Right?
is
Yeah, it's like
surprising.
they're like blood donors too, I guess. And
Such
so
such
we,
polite Canadians.
you have to, you have to. And anyway, so we basically collected a lot of the data from the literature on human blood dynamics. We tested our own with the sheep's blood, and then we started testing the viscosity and the surface tension and the density. So all those kind of physical properties
Mm-hmm.
that are really important in the way blood behaves outside of the body in terms of making blood stain patterns at crime scenes or in the lab. And making sure that it was at least close enough or statistically close enough to blood.
Do you have like it's within 5% of what when this amount of force was applied to blood, it went this distance, and when it was applied to the synthetic, it went 95% of the same distance or had the same effect or something like that.
Yeah, that's a good question. So we another interesting thing about blood and whole human blood is that those physical properties that I was talking about, they
Yeah.
actually change all the time. So they change within ourselves during the day. So depending on what kind of exercise we've had,
Oh,
what's going on in our life, and maybe you had some drinks the night before, your blood viscosity changes quite.
or right at the moment,
Um,
actually.
yeah, exactly, right
I
now.
am drinking
So
a Guinness Irish wheat.
jealous. Perfect.
So is this then the synthetic kind of like an average of different donors? I used to work
Yeah.
in blood testing, and we would get in the clot tubes. So there'd be all the red blood cells at the bottom, and then nice clear yellow serum at the top. But some people's blood wasn't like that. Other
Right.
some people had white opaque serum instead of the nice clear yellow stuff.
Hmm.
Is this then kind of like the average of what most people's blood would do?
Yeah, yeah. So we kind of modeled all of our materials based on on the average human kind of values that we saw. Yeah.
And so let's back up a little bit. And what is your degree? What's your background? In fact, let's go back further. Where are you from? And
Yeah.
where did you go to school? What was your degree based on? And tell us a little bit about yourself.
Okay. It's a lot to answer, but I will try.
All right. Well, where are
So,
you from?
um, all right. So I'm from a little town in Ontario. It's called Ayr. Believe it or not, there is a place called Ayr, AYR, and
Hmm.
population of maybe a few thousand. And so I grew up there, and in high school, I had no idea, I guess, what I really wanted to take. I knew I really liked the sciences and maths. And the classic day before the university applications were due, I had to pick. And I do fall within the majority of, I just stumbled across forensics, like the majority of your
All right. So you've listened to other
interviewees.
podcasts before. Good to know.
Yes, of course. And I took forensics. I did my undergrad at Trent University too. There's not many schools in Canada that offer programs in forensics.
Yeah.
So
If I could mention that, I've had a little bit of experience there because there was this moment where a colleague of mine and I, we almost went to go teach a semester at Laurentian every year. They were developing their program up there. Yeah, I'm sure you're familiar with Laurentian, but Trent was one of the other possible universities in the area, but it was Laurentian that was trying to bring in me and a colleague to talk about forensics because they're going to also be teaching local or professional forensic examiners as well. They're trying to do this university program for current professionals there. But you're right, at least in Ontario, you have, I mean, I've seen some interesting work coming out of Toronto. Is it Toronto University of Technology or something maybe, or something like that? And then there's Trent and then Laurentian.
yeah, so there are a few more. Windsor has a program now in forensics, and UIT or the in Oshawa has a program in forensics. And they're all kind of at the undergraduate level. And I did look into Laurentian's professional base. I think it's a master's program, but don't quote me
Hmm.
on that. It sounds like, yeah, they are teaching kind of certifications and course level things for professionals. So yeah, 10 years ago when I was originally starting, trying to figure out what I wanted to do, those programs didn't exist. So my option was basically Trent, I think University of Toronto, and I was looking at an integrated science program in Carleton. So it wasn't super forensic-y, but it had components in forensics. And anyway, so I went to Trent. So I moved up to Peterborough, and I took forensics. And kind of along the way, um, I got into actually working in an environmental monitoring lab, and we were working on looking at the way mercury levels kind of are in lake systems. And I got into developing these sol gels, like I was talking about, in order to kind of capture them from lake water systems. So these sol gel materials are really interesting because you can kind of tinker with the chemistry and get them to do lots of different things. So one thing I was doing was trying to get them to suck mercury out of lake waters, and I
Are,
got
are
really
you in
excited
the
about it.
are you in the synthesis part of it, or did the analytical part? Someone synthesizes for you, and you do the analytical after.
Yeah, I actually created the material,
All right,
and
so
then
synth.
was involved with the analysis too.
Wow, that's no,
Interesting.
usually as you're aware of there. It's usually a multi-team, multi-group. People do one thing and another. Oh, that's cool. You were able to do cradle to grave, if you will.
Yeah,
Right.
it's kind of neat. The neat thing about Trent is it's a smaller university, so you get lots of exposure, doing lots of different things.
Hmm. Okay, so
Yeah.
that's pretty cool. Now, was this all at the undergraduate, master's, PhD? Where was this work falling?
Oh, right. So this was in my undergrad, and
This
this
was
may
all
sound like
undergrad.
more. Yeah,
Wow.
No,
it was a fun undergrad.
it's insane. When I met students from Laurentian and UIT, and I was looking at their projects, and these were the undergrad or students, I was blown
Right.
away by their level of research. I've always, and I've said this in other episodes before, it's so true. Other parts of the world, in Canada, the your bachelor's or honors bachelor's and masters are like our PhDs sometimes here in the United States. It's kind of
Mhm.
sad. It's amazing what the caliber of research coming out of your universities there compared to the U.S., which is just. I did a survey online and I did a literature search. Give me a master's degree.
Even with, I mean, I went to a humongous university, and yeah, undergrads, we didn't do that kind of level. We weren't involved in the synthesis of stuff. Like Glenn was kind of saying there, other like the postdocs got to make the stuff, and then we just kind of got to test it out and see how it worked for different applications. So that's fantastic that you got to get involved so early with that.
Yeah, it was really
So
fun. I think it kind of speaks to networking and meeting real professionals too, like quite
yeah.
early in your
Sure.
career, which was really beneficial for me.
So how did all that translate from the capturing mercury to this blood application?
I know. So the story does get better, and this might be as slightly exaggerated, but this is the best way that I like to
Hey, don't
explain.
let the truth get in the way of a good story.
Exactly. So I had really got into environmental chemistry, and I was just about to kind of drop out of forensics. It wasn't. I know I had interest in other fields, and I was taking a crime scene investigation course at the time, and I was being taught by Mike Illis. And Glenn, I think you may know Mike.
Mm-hmm.
But anyway, he sent. I got this email one day that was like, "Hi Teresa, can you like meet me in my office? Want to discuss something at 5?" And I'm like, "Oh great." Like, what did I do? Did I plagiarize? Oh no, I'm freaking out because at the time he was um, a staff sergeant with the Ontario Provincial Police. So I go to his office and whatever I did, I'm so sorry. He's like, I'm just trying to offer you a
Sorry
job. I'm
about
like,
that.
oh,
Sorry about
yeah. Oh, okay. Yeah, sorry about it. And yeah, I just just want to offer you a job. I'm like, oh, well, instantly relax. Well, that's great. This is really cool. So for 2 summers, I worked under Mike at the Ontario Provincial Police as a research scientist, and I started doing research on bloodstain pattern analysis. And that was great. Like that was a completely pivotal point for me. And Mike is such a great mentor, and it just got me completely hooked onto the field and everything from like in research about pattern identification and the mechanisms
Mm-hmm.
and just what's going on in bloodstain pattern analysis really captivated me. And that kind of was a pivotal point for me in getting into research in bloodstains. And then that translated to eventually I met Michael Taylor through Mike, and
Oh, yeah.
he was looking for a master's. Yeah, so that's how I met Niki, and
Yeah.
I went to New Zealand and did my master's there, and then my project was basically making and thinking about validating or making testing procedures for validating synthetic blood substitutes or forensic blood substitutes for research and training. And so that's kind of where the idea kind of like brewed and grew from. And it's really important. I always emphasize with people when they're using blood substitutes or they're making a homemade product or something, to make sure that you have some sort of validation or some scientific backing to the product. Because depending on what you do with it, if you want to make it drip a certain way or spatter a certain way, it should be validated in some way to whole human blood or a blood analog.
Right. Right. Sure. Wow. Okay. So, and then you ended up doing your master's there in Auckland with Michael Taylor's group.
Yeah, yeah. So it was in Auckland. I did all my research down in Christchurch with him,
Mm-hmm.
and it was such a fantastic time. The it was such an eye opener too to just see how different forensics is in between Canada and New Zealand, and also meet kind of the
How
staff. And
so?
it's such a I thought most of our forensics is kind of conducted by police here and in some sort of capacity with the police organizations. Where in New Zealand, most of it is civilian based. So
Mm-hmm.
I found that very interesting. I thought it was really neat that in New Zealand, because it's such a small country, and there's only a limited number of trained crime scene investigators, that they could really go anywhere to process a scene. So some of them would be flown to the other side of the island to process a scene. And I thought that was really interesting because they had such a broad like geographical range. So they saw lots of
Sure.
different I guess scenes.
So, and then you end up getting your master's there. Is that correct?
Yes, yeah. So I got my master's in Auckland.
And now you're back at Trent.
Yes, so that I could not get away from Trent. No, actually, no, the story is better, I guess. But uh, anyway, when I was in New Zealand and I was thinking a lot about these testing protocols on testing blood substitutes, I was always in the back of my mind had this idea that these sol gel materials are even though they're typically used for things like in any sort of thin film coatings, like things that I don't know, like you can spray it kind of on anything. Most of them are created for corrosion protection, but they make these thin films. And blood stain pattern analysis, basically, when blood is deposited on a surface, it creates a thin film.
Mm-hmm.
So I found that there's a lot of overlap in kind of the mechanisms and the chemistries behind both of the disciplines. So I thought this would be a really neat adventure to pursue building a sol gel material that could be used as a blood simulant for crime scene investigation. So it was a neat kind of blend of both worlds.
Right. Okay. So
Interesting.
the development of these, did that happen here after your master's and you're back at Trent? Is that where that happened?
Yeah, it happened at Trend.
Okay. And well, I mean, did you end up getting a PhD out of all this? Because you better have. I mean, for all the work you put into this, you really should have a PhD out of this.
Well, thank you. Yeah, actually, I'm a fresh graduate, so I graduated at the end of January.
All right. Well, thank God. Good.
Well,
I,
congratulations.
Still waiting for the prescription pad with the designation of
ah.
doctor,
Oh,
but
Yeah. Okay. Well, then we should give you your proper title, Dr. Stotesbury. I didn't I didn't know, and that's good. I mean, I'm just listening to how much work you put into this, and I thought, my God, if she ends up with, not that there's anything wrong with it, but just a master's out of all this work, that it just, compared to what I see again, some people in the United States getting their degrees. I mean, okay, so I'm happy. All is right again. Okay. And now that you're back at the university, what are you doing now that you've got this great, blood substitute that you can sell? I assume you guys have a patent at least submitted. What are you guys doing with this? What's next, and what's your next steps?
okay. So we are commercializing our material. We basically have decided that we've formed a research group called Impact Scientific, and we are creating lots of the materials for training and also, interestingly enough, high school education. So
Hmm.
oh yeah.
yeah, so we noticed, I found this really interesting paper in an education journal that said that it was a study conducted in 2016, and out of 407, I think, students interviewed, they asked about a bunch of different professions, and they asked the students, do you think that these professions require math and science to do your career? and one of them was a forensic analyst. And they reported that I think it was something like 40% of students that were asked were unsure, and 29% of the students said, no, you don't need math and science to become a forensics analyst. And it kind of blew my mind. I thought, well, we need to change this perception, because there's obviously you need lots of math and science to become a forensic analyst. So we took basically the dripping experiment. So in order to do a basic bloodstain course and get certified, one of the experiments that practicing bloodstain pattern analysts will do is look at the way the size and the shape of a bloodstain changes with increased dripping height or impact velocity. And normally the stains get bigger, right? And you get lots of little or edge characteristics like spines and scallops. Those numbers increase with increased impact velocity. And they get into this idea that everything grows until the droplet reaches terminal velocity. So we kind of took that experiment and condensed it down to a grade 11, grade 12 high school level, and I've been part of my month of mayhem has been going out to high schools and demonstrating the kit either in a classroom full of students or with high school teachers, and it's been going over really well. But in a friend, yeah, it's quite interesting. It's neat to kind of blow that myth that crime scene investigation is so glamorous and it's like the Hollywood kind of facade.
Right.
It's neat to show students that like this is a serious and a relevant career option that you could take if you keep science and math and engineering and things like that in your future.
Do you guys let them play with like a hammer and do some spatter?
Well, we were a little bit nervous. Well, I was mostly nervous about the blood, guts, and gore. So with students, so we basically just have them making circular passive drip stains with a little pipette. So I'm sure some of them really want to, but we kept it pretty kosher.
Okay. Oh yeah, you just get out onto the football field,
Right.
And maybe some sort of portable tent device to maybe keep it a little contained or off anyone's clothes or something. But they'd probably have a field day with that. They would love it.
Yeah, it's been going over really well.
So one of the things that I think Niki had told me about was that you guys had added probably fluorophores in some way to your molecules, and that by adding these fluorescent tags to it, you could change the wavelength of emission so that you get certain colors. So some of your blood substitutes could, under an alternate light source, might emit and appear red. Some might appear yellow or orange, which allowed you guys to differentiate which blood droplets came from which impact patterns. Is that right?
Yeah, and this is actually something I think is really important to pass out to your audience. So I'll talk about it now.
Let's talk about that, because I found that absolutely ingenious.
Yeah, so I would love to talk about this in depth. Okay, so analyzing complex bloodstain patterns can be quite difficult, right? And especially when you have patterns overlapping and it's
because all the blood looks the same.
All the blood looks the same, exactly. And what's even more difficult, and just through conversations with bloodstain analysts at different conferences or just through contacts, it's always very difficult to sometimes get someone to train someone in disentangling complex bloodstain patterns. So how do you tell someone there's lots of impacts here, or how can we start teasing out sequencing and things like that. So, our research group thought a lot about disentangling these complex patterns and how we could kind of address that as an issue in training. And the beauty of building artificial fluids or building blood substitutes is that you can really add anything you want to these things as long as they're behaving the way they should be, and I guess behaving like blood. So we thought, well, there's a Center for Teacher and Learning at Trent, and they work a lot on spatial reasoning. And through conversations with them, we learned that you can disentangle complex spatial kind of images through color. So that's one thing that can help a person learn and kind of understand and figure out what's going on. So we thought, well,
Amazing.
this is easy. Why don't we put colors into these blood substitutes? So our original thing was for complex bloodstain patterns, let's put some colors in there, and then we can, you can make a yellow impact pattern, a red impact pattern, a green one, a blue one, and you can play with things like creating multiple impact patterns at the same area of origin. So you can see how what's really important to say when you're testifying or writing about is saying a minimum of one impact, because the physics, you can have multiple impacts at the same point in space, but when you're doing your trajectory analysis or coming back to that area of origin, it's not going to tell you how many impacts were there. It's just going to tell you in a three-dimensional coordinate in space where that blood source was. But it gets the point across that, hey, these stains all look the same, even though they were made from multiple impact events at the same area of origin.
Yeah.
And you can, it's quite interesting, and we do things too, like you can move, move kind of the X value, or we call the X value, and that's the distance of the blood source from the wall. You can move that back out, and that changes a lot of the shape characteristics of the blood stain, so that ellipse that you'd see. And it shows how the patterns change with changing X's or distances from the wall, in all with the same kind of Y and Z's, Y being the distance from a fixed point in space, so left or right, I guess, on a wall, and Z being the height value. So it's really neat to look in color, and you can really start in training programs, start looking at these blood stains and start picking out limitations or picking out learning points to talk about with students.
And what is it that would activate the colors? What specifically, will cause it to fluoresce, glow, or whatever?
Right. So the ones that I was just talking about, they're colors in white light. So we just add different dyes. But yeah, we took it one step further by saying, all right, well, we all bleed the same color, so you need the next point in learning. And so we created these substitutes where, they're all red and white light, and then we've added phosphorescent powders into them, and they'll emit, they'll glow at different colors when you kind of shine UV light on them.
Cool. Yeah, that's.
Wow.
I mean, that to me is just amazing, because then, having gone through these kind of practicals, you sit there for hours and hours, and you determine your point of origin and the mechanisms creating these different patterns. And then, of course, you debrief and watch the video that someone made of making those patterns. And that's the traditional way of learning. Oh, okay, so you got hit 3 times, and you can see that happen. And but the way that you described it allows for such a visual mechanic of seeing which stains came from which impact and which mechanism, and maybe, maybe in a realistic, like a homicide scene, the adrenaline's up, those first few hits are maybe, the most forceful, and then perhaps the force starts dropping off because they've been wrestling, or, it's hard beating someone to death. And so maybe those last blows don't have the same energy, so you can see what happens to the stains, and you can see with these colors, these are from different ones and they reacted differently. That must be just mind-blowing to see that kind of reveal at the end.
Oh, it's just so fantastic. And we're doing a lot of research now to kind of quantify we're hoping to do a lot of research right now to quantify and qualify the learning from this, and also engagement and perceived benefits of the technology. But another thing, it's just so fantastic because what we do is we do a lot of learning off images. So I'll actually photograph these complex blood stain patterns when they're glowing in the dark. So then trainers can go in and work off the image. So you can blow up these patterns if you're using some sort of, I don't know, digital projector or something like that. Or maybe every student can actually go on their computer and actually zoom in and out of these patterns. And you're right, look at particular stains or certain areas in the pattern where they want to figure out, okay, well, this came from this mechanism, this came from this mechanism, and how is this going to help me when I go in to start processing my scenes? And what I think is so neat, I had this really interesting pattern where I think I made something like an impact pattern and say something that glowed blue, like a dark blue, and then right after it in sequence, I put some orange of the synthetic and made a handprint or a contact transfer, and kind of not only the transfer but also wiped the blood across that pattern. And so you can see the blending of the colors. And it's so interesting to start thinking about those mechanisms, but the kind of further point of that learning is now you can start adding different components of your crime scene analysis. So what are you going to do with blood? Well, you're probably going to swab some areas to figure out who the bleeders are. And visually, when you see these mixings, like it helps maybe spark thinking about, okay, well, where are appropriate areas that perhaps I should be swabbing from, or thinking about analyzing further.
Right. Yep. So have you guys discussed or thought about adding amino acids to your to your blood substitute, which could open up a whole series of dye stains being applied to them and other kinds of visual inspections that crime scene investigators have to do as well?
That's a great question. So right now, one of our fillers in our newest products has an iron additive in it, and so it will react with peroxidase-based reagents.
Sure.
So we're actually in the
Glucocrystal
process
violet.
of a master student. Yeah, and luminol, which
Yep.
is fantastic. So we have a master's student trying to optimize that. And yeah, and you can imagine then the next thing is to add amino acids in. We also have, also researching right now adding DNA into our fluid to
Oh,
use it for
wow.
training or use it even as like a lab standard. So no longer do you have to use someone's blood, I guess, as a control. Maybe you can use
Sure.
ours. Yeah, so it's really neat because the material is so versatile that you can add lots of things into it to get it to do all these little bit of enhancements. But because we're all scientists and researchers, these things take so much time because we care so much about the details and making sure that everything
Right.
is
Mm-hmm.
well, you guys too, right? But making sure the details are all there and all the tick boxes are checked. So we decided that we'd keep everything in this program or a research program for research. So anything that comes in, if people are buying the material or using it for their training courses, anything that they can contribute back to us will feed into building the material to be even I guess stronger in a forensic relevance.
Does it have an expiration date or degradation? Is it kept at room temperature? I mean, those are the things I'm thinking about with blood. You always have to, worry about those kinds of issues. But anything like that with this is pretty stable.
Yeah, so I can tell you a little bit first about why the motivation behind making the synthetic blood substitutes. For sometimes, as I mentioned, it's difficult to source blood sometimes and get it into labs. Sometimes it's difficult to obtain the ethics approval to actually, again, bring a biological fluid to your lab.
Sure.
But working with blood is really tricky because you have to keep it at a physiological temperature, so 37 degrees, and our blood is meant to clot when it's exposed to oxygen. So that's why we have anticoagulants in blood, and it's sometimes hard. Yeah, it has a very limited lifespan when you're working with it. So I think we'll use it for around 2 weeks, no more than that. But we try to do everything within the day or two that we actually obtain the blood. And what's great about, and also the red blood cells will hemolyze and break up, and the properties of the blood will change over time
Mm-hmm.
with age. And there's some research that show that in the literature, which is quite really interesting to go in and read. So our material, it's stable at room temperature, which is fantastic. And I've had some of our material spinning and remain liquid for over a year, but we've only really
Oh,
been
wow.
testing things for a month long. Yeah, it's quite exciting. So I've only really been testing for things in month-long timescales. Um, but what's kind of neat too about our materials is that they can be rehydrated too. So that although they're catered to dry and simulate drying and clotting of blood, you can kind of rehydrate them too to get them to stay in the liquid
That
form.
is so cool. So cool.
It can do anything.
Oh, but it's so exciting. But now I need to go in and get these numbers. So like we've simulated clotting and simulating drying, and now we need to go in into the research more deeply and figure out, well, what's the mechanism of the drying and the polymerization. So it's should be a really lifelong project, and I think that that's so exciting, and it would be neat to have more
Yeah.
people involved and working on it. So yeah, I'm really quite proud and excited of with the project.
So along those lines, uh, it seems to be, just like you said, it started with this, mercury removal from lakes, and now it's a blood simulant. What what other, things, do you imagine this sol gel, might be able to be utilized for, in forensics specifically?
Oh, great question. So remember how I was telling you guys that we could put DNA into the material, or we're just working on that?
Right,
Hmm.
right.
Well, the next kind of extension to that is using it as an environmental modeling or monitoring tool. So you can have the DNA, or you can bind it to the colloids and put it into these lake water systems or any other systems, and get specific markers to kind of figure out, well, what species of wildlife, or I guess in other,
Oh, wow.
yeah, situations, I guess depending on the environment that you put it in, what DNA is out there from what source. And I think that kind of yeah, that's the next kind of thing, I guess, for us that could be really interesting and has like a broader outreach to not only forensics but things like environmental monitoring
Right.
and
You can like monitor for invasive species, or yeah, you could really open up a lot of stuff with
oh, it would
that.
be so fantastic. So that's our pipe dream at the moment. So we're still working on it, but that would be really
That's where the
neat.
real money is, not forensics.
Yeah,
Exactly.
I must say it is quite difficult for us up here in the Great White to find funding for forensics. Um, we're quite limited, but we still manage to get by.
So, Teresa, just curious, will we see you at the IAI? Will you be peddling your wares there? Will you be getting a booth? And because obviously that's a great conference, the International Association for Identification Conference, because there's so many bloodstain pattern analysts that go there for training, and some of the world's renowned experts. That would seem like a great place to market this product.
I wish, Glenn. I'm really sorry. I had to choose between that or the IFS conference in Toronto. And
Uh
I
huh.
am actually giving, along with Mike and another colleague with the Ontario Provincial Police, we are giving a workshop on what I've been talking to you about with the colored blood substitutes, the glow in the dark blood
Sure.
substitutes, and other pedagogical like technologies and blood stains at that
Sure.
conference in Toronto this summer.
Well, so that conference is every 3 years, which means we'll see you at the next two IAIs then.
Yeah, you bet. For sure.
All right. Well, Teresa, thank you so much for coming on the Double Loop podcast with us. Is really interesting topic, and exciting to discuss kind of where these things are heading, and I don't know about Glenn, but my mind's kind of spinning with possibilities of,
Absolutely.
of where this
Absolutely.
kind of stuff might lead.
Oh, thanks.
So yeah, thank you so much for coming on, and we'll have to send a special thanks to Niki for for suggesting that you come on, and making this all kind of happen today.
So, Teresa, before we let you go here, can you talk about any of the upcoming training that you have either, well, training that you've already done internationally, or maybe even training that might be coming up that listeners, if they want to get involved in this or see this product in use in training courses, where can they go to take some workshops, or how can they get a hold of you to maybe bring you and your crew to, if they want to host a workshop or training event?
All right, so we were contacted from a program that was running a Bloodstain course in Algeria. So Liz Williams and Rebecca Book contacted me, and they had seen we put out a paper in the Forensic Science Policy and Management Journal that talked and discussed a little bit about the colored blood substitutes, the glow-in-the-dark blood substitutes, and how and kind of very briefly how spatial reasoning works and how this could be a really valuable tool in training. So they had contacted us about bringing in some of our technologies into their training course. And what was quite neat about their training course is that they took a 40-hour course and completed it over the course of 3 weeks. And they did this basically to get more hands-on training and more kind of mentorship in their training. And so what we did, so we made some patterns, some colored patterns for them. We gave them some of the material to make their own colored blood, and then we implemented, or we gave them some blood stain patterns that were all red in the white light, some complex patterns, and that glowed in the dark. And so like I'd mentioned, we got them to kind of work off the images and kind of look at the glow in the dark patterns. And apparently the feedback was quite positive. So I wasn't at the training course, but I did supply the training materials, or a research group did, which is quite interesting. And it was so neat to kind of beta test that technology in an actual training course. So there was some pretty good positive feedback about that. And I think you're right. I think that when you see these patterns in color or in the glow in the dark, you just get this wow moment where learning starts and discussions kind of spawn from that. So that was quite exciting.
Someone wants to get in touch with you about any kind of this stuff.
Right. So we're going to be hopefully running a workshop down at the IBPA conference in September in Redondo Beach. So hopefully stay tuned for that. So if any bloodstain pattern analysts or anyone going to that conference, make sure to touch base with me there. You can look at what we're doing in our bloodstain world by looking at www.impactscientific.ca, and you can either get a hold of me there, or you can contact me at Trent University. So you can contact me at Theresa Stotesbury at trentu.ca, and I would be happy to help with any sort of questions that you have, or trying to get forensic blood substitutes into your research and training. I love collaborating, and I think it's so powerful when you have practitioners and academics pairing together to kind of figure out more about forensics and provide answers to lots of different questions. So make sure to feel free to contact me there. I would love to talk to you.
So that's awesome. All right, thank you again so much, Theresa. You can always, as always, contact myself, Eric at rayforensics.com, or Glenn at eliteforensicservices.com. And then keep listening on Stitcher, SoundCloud, or on iTunes, and we'll talk to you guys next time.
Bye, everybody. Have a good week.